Hello everyone, my name is Nelson Serre and I'm currently a postdoc in the RDP lab in Lyon, France, and I'm working under the supervision of Yvon. And I would like to thank the organizer of this webinar for inviting me to do this talk today. It's a nice opportunity, and so today I'm going to talk about microfluidics for plant root biology, a tool to decipher rapid and dynamic responses. So how did I got there talking about microfluidics? I basically had a biological question that required to observe the very fast effect of treatment in the magnitude of seconds to minutes. And so I needed a way to switch from a control medium to a treatment medium without stopping the microscopy imaging, but also a way to normalize my basal staining intensities to a control level. And so microfluidics is basically just working with fluids in tubing and channel that are less than a millimeter, so in the micrometer range. And it's a range where fluids, it's a little bit more predictable because you can have something called laminar flow happening. Fancy name, but it's just basically if you have particles, or here we have ducts in this example, flowing into a channel. If you have laminar flow, they will flow each along the side of each other without mixing. And if you increase the size of this channel or tubing, you might get something called turbulent flow, where things starts to mix and be a little bit more chaotic, less predictable. And for more live example, let's say, you can actually achieve laminar flow in bigger tubing than a millimeter, like your faucet, your tap water faucet. When the water is like crystal clear, there's no turbulence, no air inside. This is laminar flow. And then on the image on the right, you have perfect example of turbulent flow where everything is chaotic. It's very hard to predict. Everything is mixing fast. Here you have even air mixing in there. And if we go back to microfluidics and a microfluidic chip, so a very simple microfluidic chip, you have two inputs with a medium in yellow and a medium that is green. If you have laminar flow and they go in the same channel with the same pressure, they will not mix, they will just flow side by side. There will be a little bit of mixing by diffusion, the interface of the two medium. And if you actually change the pressure of one for another, you can actually play with the beam of one and the other. And if you enlarge the channels a little bit, or a little bit more even, you will get the turbulent flow, which has all these vertices and chaotic behavior. But this actually is not a bad thing. Like, you might want to mix something fast. So microfluidic people play between the size of the channels to obtain both of these properties in different situations adapted to their needs. And so in general, microfluidics allows for more precise liquid manipulations. And so today, after this little introduction, I wanted to do a non-exhaustive root focus history of microfluidics, and then I want to show you basic blocks to build a microfluidic system. But just for context, so I'm a plant root biologist, and I work with a plant model called Arabidopsis thaliana, which gives plants at mature stage that are like 20 centimeter high. So it's quite a small plant. It's quite small plants, yeah. And at the root system, so for example, after 14 days, you have a primary root, which has secondary root 2 branching on this one. And then a more mature stage, you observe like tertiary, quaternary roots, which gives a complex root system in the soil. But to be honest, in the lab, most of them study them at this stage, which is 4 or 5 days old. They're growing on agar plates with plant-specific medium. And here we can only see the primary root, which, all the roots of Arabidopsis thaliana are very nice for microscopy because they're about 100 microns large. They only have a few tissues, and they also have zones, which we know how to recognize them, which are composed of the root cap, which protects the root when it goes into the soil. Then you get something called the meristem, which is the dividing zones of the root. and the cell kind of queue in lines after one another until they reach something called the transition zone. And it's the transition between division and elongation. Well, cell will gain in length until they reach the final length, and they will enter the differentiation zone, which is a zone where you can start to see, for example, root hairs, which are important for nutrient acquisition. And so for the history of microfluidics for plants, actually quite recent. The first mention was in 2010. It's a very complicated chip in manufacturing and also to prepare, I think. They managed to put a root transversally to a microfluidic channel, which has for input one control medium input, one other control input on the other side, and in the middle you have a small channel which is filled with this green dye, but it could be a treatment. And basically by playing under pressure in these two control channels, you can play on the size of the beam, the green beam, and then you can treat, for example, a cell or 1-2 cells or a full tissue. And so this allows, by exploiting laminar flow, to perform localized treatments. And then in 2011, so just a year after, the chip that really democratized plant root microfluidics is called the root chip, developed by Guido Grossman, where they grow the plants in cut pipette tips filled with agar. So they put the seeds there, it starts to germinate, grow in there. And when it reached the end of the tip, they put it in these microfluidic chips, which are made with kind of like a silicone rubber transparent polymer, which is called PDMS. And then the root can find their way into microfluidic channels. Sometime, in this case, they don't want to go in the channels, they go back up. But then you can inject whatever treatment you want and do microscopy imaging at the same time. The next year, something called the root array was published, which was made to do live imaging of gene expression. So they have an high number of wells where they grow the plants, and they all express fluorescent biosensor to study gene expression of a high number of roots over time. Then I put this example of chip, which was defined as a high throughput root phenotyping chip. And I don't want to offend anyone, but there's no such thing as high throughput in microfluidics, except if you have like 2,000 chips at the same time. But anyway, then there was an iteration on the root chip, which is called the dual flow root chip, which really exploit the laminar flow property to our advantage. The root is growing into a channel that has these small pillars that allows the root to be exactly in the middle of the channel. And then what you can do is that you can inject, for example, a control on the left and a treatment on the right, or two different treatments, and see if there's different effect on both sides of the root. And a more recent one, they use the same technique to grow the root in the chip. So with this pipette tip filled with agar. And then the root finds its way to a big chamber where they inject bacteria, and then they can look at what's happening at the microscopic level between bacteria and the root. And then I'm going to talk about a little bit more about my work. So I'm going to be a little bit biased towards this chip that I developed. So what I didn't say is that this kind of chip is bonded covalently to glass. So it cannot be reopened. Most of the time when you're done with the chip, you have to throw it away. So what I did was to not bond it to a cover glass and fill it manually. So here you have a chip with two channels, two input that is empty with the channels in orange. We put a drop of medium, like in green in there. We spread it manually, and then we can transfer plants that have been growing in usual conditions, so in agar plates. They are 4 or 5 days old, so we don't need to grow them in the chip, which is very convenient. And then we put a cover slip manually, and then it goes into this contraption just so there's screws just to maintain the temperature so there's no leaks. And so, yeah, I forgot about that. That's the actual chip in a picture, which is kind of annoying to take in picture because it's transparent. And then we mount it, in our case, because we want to image the root in their orientation to gravity, the natural orientation to gravity, into a vertical mounted confocal, which is just a regular confocal, but it's put on its back. And then, so the main advantage of this technique is that you, at the end of your experiment, you can just reopen everything, wash it, repeat the experiment. And so that would be kind of my typical afternoon. I'm doing about 5-6 chips per afternoon. Instead of one that it took me 1.5 weeks to grow the plants in. And so for me, the microfluidic pros and cons for root microscopy is that first you have internal control for each plant because you image the root both in control then in treatment, which can help with reproducibility and normalization of like stainings or basal root growth, this kind of thing. Use less reagent because you work in microfluidic range tubing. For example, if it's toxic, expensive, etc., you have access to the very first seconds of treatments. It makes quite beautiful movies that you can quantify, of course, after. And the chips and even the systems are mostly limited by imagination. You could think of anything. You can also include gas channels in there, temperature sensors, all these kind of things. But there's a lot of designs already available for roots, but also for any kind of living organism. And then, yeah, if you want to automate your system, it's rather easy with fully commercial systems. But on the con sides, it can take time to master because it's a lot of, it's a lot of new stuff, basically. And on the con side, you have also small bubbles which can clog your channels, big bubbles which can completely clog your channels and move your roots, this kind of thing, clogging also. It's a lot of standard brands and vocabulary, but it's similar to any technique you will learn. To make the chip, you might require access to a clean room. I will talk a little bit about chip manufacturing after. And if you don't have a fully commercial system, you might need some basics of electronic coding to make some tinkering and manage full automation. So to fabricate the chip, you can go, there's several ways. I will talk about easy stuff first. You can use resin 3D printing. So basically you model your mold in CAD program and just print it. Then you pour your PDMS in, so your silicone-like rubber. And then your mold and your chip is ready. The problem is that with resin printing, it actually leaves some inhibitors of PDMS curing. You can avoid it, but it's a bit annoying. And what you can find is also, there's a brand called CADWorks 3D, which sells commercial microfluidics printer with their own commercial resin, which apparently works the first time with no treatments or anything. You could do micromilling, so either by micromilling metal, then make an impression of PMMA, which is close to acrylic. And then you mold it, or you can directly micromill some acrylic and mold it with PDMS. Some methods are a little bit more tricky. You can use tape, like for example 3M, the famous tape manufacturer. They actually sell tape that have defined thickness, like 100, 200 microns high. And you could use something like a Cricut, which is basically an automated cutter thing to cut your design. But you could use also fishing lines, which has a very defined diameter, or 3D printer extruded filaments, which will give you, for example, for a 0.2 millimeter nozzle, a 0.2, more or less a 0.2 filament. And then there's some tricks that have been developed by Vittorio Saggiomo. I hope I'm not butchering the name. And I can highly recommend his YouTube channels and academic work because, for example, he developed a method to make this kind of twirly things into the chip where it's basically just ABS, so plastic used in filament printing. And then this ABS is actually soluble in acetone, so you can mold with the filament of ABS and then dissolve it, and then you only have the pattern left in the chip. But the historic method and one of the most used today still is UV photolithography, where you have a substrate on which you put a photoresist, which is a resin polymer, on it. You spin it at a defined speed, which gives you a defined height. So, for example, the height of your channel. You expose it to UV light with a photomask to only expose what you want. So, for example, your channels. Once it's cured and developed, you obtain your channels in positive in this case. And then basically that's your mold, and you can just mold your PMS and mold it. And in this case, bond to a glass substrate. And so for a little bit of imaging, that's you don't have to do fancy imaging to have access to interesting information. For example, I've done tons and tons of this kind of experiment. It's in bright field. I'm just looking at the root growth and the effect of a treatment. And here it's the effect of a peptide called RAF23, which completely stops roots elongation in like less than a minute. As you can see here, when the time is negative, it's controlled, and positive, it's the treatment. But then you can image also very fast processes. Like here, you're looking at a LUT that is reflecting a membrane polarization dye reporter. And it's the effect of a phytohormone auxin, which depolarizes the root transiently, and most of the effect is actually found in this zone. Can I replay it? Oh. Yes. So here it's going to increase in fluorescence in a very fast way. Then you can also do some more play with the liquid flow, basically. So here what you're looking at is a pH reporter of the medium. The root is magenta, but it's just the LUT. It's actually not stained. The more magenta it is, the more acidic the medium is. So it's a relative information. And the more green, the more alkaline. And here, at first, I'm imaging with flow. So I'm imaging more or less the pH of the medium that is in my tube. And then when I stop the flow, the root starts alkalizing the medium in like a few seconds. And then more recently, so now I'm working with phospholipid biosensors and these RAF peptides. And you can do subcellular imaging basically and see that when you put the RAF23 on this biosensor, the plant expressing this biosensor, you can see the signal leave the plasma membrane to get into these intracellular compartments, which are, among other, endosomes. And a last example, I really like this one, is the effect of sodium on the storage of potassium in the roots. So when you put sodium on roots, they actually kind of leak all their potassium from their cells to counterbalance the massive entry of sodium in the cytoplasm, and kind of like temporarily at least reestablish their membrane potential. This biosensor is a ratiometric biosensor, and the more green you see, the more potassium there is, and the more magenta, the more the less potassium you have. And when you put salt on it, you have both an effect on growth, so the root is actually shrinking, but you also go from full green, almost full green, to almost full magenta. meaning that the root have lost most of this potassium. So let's go a little bit more technical now. So the goal of microfluidics is to push or pump liquid wherever you need it. And as I said, everything in the range of the micrometer for microfluidics. So to push the liquid, literally you can have the liquid into a syringe and either operate it manually, which was done in the Masala paper, or you can use automated syringe pumps. But you can also pump the liquid from a container to your chip using peristaltic pump or double diaphragm pump, annular gear pump. There's a lot of type of pumps that I'm not going to describe all of them today, but just to give you an idea. And then what seems to be a very nice way to control microfluidics is pressure-based microfluidics. Where basically you have a source of compressed air or anything compressed, but compressed air is better for working into a microscopy room. You can have a manually controllable pressure gauge, plus maybe a solenoid, so a valve that is controlling on-off the flow of air. Or you can have something a bit more fancy, which has electronic control in it and feedback, so you can set a defined pressure and push your liquid at a defined pressure in your system. And then you can find fully commercial designs based on these pressure controllers. In Europe, the two competitors are Elvaflow and Flugent. They both have more or less the same produce. The maximum output channels are 4 in both cases, and in both cases they have software and automation capability through like some very easy macro, and you have all the associated device like flow sensors, valves, bubble traps, and other things. And I will not talk so much about it today, but it's possible to add elements from other brands with some tinkering with like microcontrollers and things like that. Then you might want to control where your flow is going or when is it going somewhere. And for that, you need valves. So you can use manual valves, either like just on-off one or a distributor valve. You can have it electronic controlled. So like these little ones, I use those ones, which are from the brand Memetis. You can also have a more distribution valve with like 2 inputs, 1 entry, or even this rotary valve with a lot of outputs. And then you might want, and I will explain why, measure your flow speed. So for that, you need flow sensors. And to my knowledge, but there might be some others that are very common. You have thermal flow sensors, which basically you have 2 thermal sensors and a heater in the medium. It will heat the medium a little bit, and by analyzing the trace by some math that I don't have any idea how, you can actually deduce the flow in like microliter per minute. And then there is also something called a Collier-less flow sensors, which is based on vibrations. And that I will not be able to talk about because I really don't know how they work. But they are supposed to be more precise. So why would you bother with flow sensors? So basically, it's really, in my opinion, maybe I'm completely mistaken, but if you work at constant pressure and you have a channel that is fully blocked by a bubble or a clog or something, there will be no flow in your channel, but you will actually don't know because you have no output of this information. While if you work at constant flow, so if you mix pressure and flow and their feedback on each other, the pressure will increase until the bubble is displaced, or you reach the maximal pressure of your system. But at least you will know that it's actually blocked. And if you have a partial clogging, kind of the same thing. At constant pressure, you will have lower flow than expected, but you will not know. Constant flow, you will be able to increase the pressure until the set flow is reached. And so to talk a little bit about money, you could have very minimalist system like syringe that don't cost anything. PDMS on its own doesn't cost anything. You could have a system and cheap production for way less than 500 euros. But then if you want something fully commercial with all the device associated, it can be more than 10,000. But I mean, given that I'm talking to microscopists, these sums are quite okay compared to microscopes. And so in my opinion, a microfluidic system is a compromise between a lot of parameters, such as need, cost, precision you need, and if you need stability and automations through software or electronics, but also the time you're willing to put into its development. And so to finish, I wanted to show you my example of a microfluidic system, which is a fully automated system which has continuous flow. So by continuous flow, I mean that I have two pressure outputs that pressurize two tubes. One is my control, one is my treatment. And wherever they go in the chip or not, the flow is always moving to avoid sticking of things, precipitations, and other things. And so I have these pressure controllers that control the pressure in millibars that is connected to the microscope for automation, but also to its software. And to show you that it's possible to tinker a little bit and add what you want, I also have a macro pad which triggers some macros of this in this software, and also a valve controller because I did not buy the valves that Airflow was selling because they were not convenient for me. Then, so this pressures my two tubes that goes then to a device called a flow sensor, which gives me the flow in microliter per minute. These two communicates to maintain the flow at a dedicated, at a specific flow rate. And then it goes into something that I call the valve highway, microfluidics highway exchanger, where it's the continuous flow part things, where there is basically, it's a one entry, two output valve system where the liquid enters in the middle and then it kind of goes towards the trash or towards the chip, and that for both lines, they're identical. And then the liquids are going into the chip, which in my case contains two inputs and two plans. And then I already say that, but we mount the chip on a vertical stage microscope. And so to give you a more dynamic idea of what's happening, when I image in control, my control is going towards the chip, but not towards the trash. And my treatment is going towards the trash, so it's always moving, especially not staying at the flow sensor, which has heating element, but it's not going towards the chip. And then I have macros and also my macro pad I show you where I press a button or I can adjust time in the macros to trigger it automatically. Just to change this combination, so the control is now going towards the trash, not towards the chip, and the treatment is slowly replacing the control medium in the chip. And so on that, I'm done for today, and I would like to thank two persons, Dr. Matthias Fendrik, which is a PI in Czech Republic, which under the supervision, I did my first postdoc under his supervision, and it gave me the bug of microfluidics, basically. And actually, for the story, at the beginning, I didn't want to do any microfluidics, but now I'm basically only doing that. And then my second postdoc supervisor, Yvon Jaillet, which entertained this bug and allowed me to learn even way more. And also, of course, you for your attention, and may the flow be with you.